Actually, the title of my talk could have been, let me see here, Genetic Mechanisms of Susceptibility to RSV Disease, which is in fact what I'm going to be focusing most of my talk about. So my collaborators and my group over the last few years have adopted a systems biology approach to understand complex diseases. And this is an interesting and I think nicely summary of a systems biology approach, in this case for cardiovascular disease. And what these boxes represent are the genomic approach or genomic tools, transcriptomic tools, proteomic tools, metabolomic tools, and finally phenome. And all of these sorts of approaches interact to help us understand the etiology and mechanisms of susceptibility to complex diseases. What I'm going to speak about today are some genomic and transcriptomic approaches that we have taken to understand some subphenotypes and disease related to RSV infection. So how do we understand the genetic mechanisms or the genetic basis of susceptibility to RSV disease severity? Over the years, traditional family-based studies have been used to understand complex diseases. Twin studies have been used, and more recently, genome-wide association studies, or GWAS, have been used. Association studies, of course, are very useful, but the genes that you study must be selected very carefully, largely based on biologic plausibility. And then animal models have also proved to be very important and helpful in understanding human disease. As I mentioned, expression array studies have been used, and genome scans, similar to the GWAS in humans, have also been used to help understand complex lung diseases. And I'm going to focus now on some work that we've done using genome-wide approaches in inbred strains of mice. So this is a schematic of sort of the approaches that we use in my laboratory to help us understand complex lung diseases. We take advantage of a large number of inbred strains of mice that can be used for investigating differences between strains to help us understand the genetic basis of susceptibility. We can also use human cell lines, and both of these approaches together, using global gene expression analyses, expression QTL analyses, and then haplotype association mapping, which are all gene discovery approaches, so they have no a priori sort of basis for selecting genes. We can identify candidate genes and networks using these integrated studies. We also consider biological plausible candidates in our analyses to enhance our profile or gene networks. Ultimately, once we identify these gene networks, we test the mechanistic sort of validity of these genes, and then test them, these gene candidates in human populations doing epidemiological studies or clinical studies. So for the remaining time that I have, I'm going to hopefully tell you about 3 separate studies that we've been doing related to RSV. The first study is a murine RSV disease model. The second is RSV disease using HapMap cell lines, and I'll explain what HapMap cell lines and how they can be used later. And then if I have time, I'd like to talk about RSV disease and toll-like receptor 4, which is a study that Fernando's group and mine have been working on for a number of years. So the first study. So what are the genetic determinants of susceptibility to RSV disease in mice? This was a project that was undertaken by a graduate student from the University of North Carolina, Monica High. And Monica took advantage of the extreme genetic diversity that's represented in this phylogenetic tree. So these are nearly 100 inbred strains of mice. And you can see that there are 7 different groups, 7, 6, 5, 4, 3, 2, 1, and their relatedness to each other based on the phylogenetic tree. Monica chose mice that are outlined here in the gray boxes, from each of these branches to represent the greatest diversity that we could possibly get in the mouse. By previous studies, have found that 30 or more strains are really optimal for doing these kinds of association studies. So she chose 30 strains, and she infected the strains with RSV19, RSV19 or vehicle, and 1 and 5 days post-infection, mice were sacrificed. And we performed bronchoalveolar lavage for inflammatory cells and total protein. We also measured intraepithelial cell mucus and RSV-N mRNA as our phenotypes for disease. So this slide shows you 2 important points from all of the work that she did. So what I've plotted here on the left panel is the number of PMNs, or neutrophils, that we got in lavage after vehicle and days after infection. And what you can see is 2 different strains of mice, BALB-C and C3H-HEJ. And BALB-C, you can see, has a very robust response 1 day after infection, which is attenuated or resolved by 5 days post-infection. On the other hand, the C3H-HEJ mouse has a very reduced response relative to the BALB-C. In the other panel, we have monocytes on the y-axis. Again, the x-axis is the same. Monocytes don't peak until 5 days after infection and are resolved about 10 to 15 days afterwards. And again, there is a marked strain difference. So the BALB-Cs are very responsive and the C3Hs are very non-responsive. So the 2 points, the kinetics are different, and there's very significant interstrain variation in the response to RSV. So this is the results of all the strains of mice. What we have on the top panel are the monocytes. The next panel are lymphocytes. These are PMNs, and then this is the RSV mRNA expression. And these are all the different strains of mice that she phenotyped in her response to RSV, and they are in order low to high according to the monocytes. So these are very high responders at this end, and these are very low responders at the other end. So 2 points that can be made from this. There's significant interstrain variation, as I showed you in the previous slide. Across these 30 strains, there's a tremendous range in the amount of response that we get to RSV. Another important point is that this is a continuous distribution. And what this means is likely there are multiple genes for each of these phenotypes that contribute to this differential responsivity. And the final point is that there's a lack of correlation between the phenotypes. And what that probably indicates, and I'll show you evidence that suggests that it is that there are different genes for the different phenotypes. So immediately you see that this is a very complex model. We likely have different genes for the same phenotype. The phenotypes do not correlate with each other, which indicates that there are different genes for each of these different genotypes or phenotypes which interact in some way that we don't completely understand. So we can take these phenotypes, each one of these, plus others that we accumulated and haven't shown you, and ask whether we can locate the genes that are responsible for this differential phenotype in the mouse genome. And we do that by haplotype association mapping. And we use a number of algorithms to ask this question, one of which is called SNPster. SNPster has 650,000 SNPs that are mapped in the mouse genome. And at each locus or haplotype, we can calculate a genotype-phenotype association. I'll explain that again in a second. And then through this association, we generate log p values for the association of the phenotype with each of these SNPs or haplotype, and then plot them across the genome. So here's how this works. So these are the strains of mice that we phenotyped on the top. These are the SNPs, and these are the genotypes of each of these SNPs for each of the strains. And this is from the proximal end of chromosome 1, and we do this all the way to the distal end of the Y chromosome. And so the way this works, we take a 3-SNP window, and consecutively down the entire genome, we ask whether there's an association between that 3-SNP window and the phenotypes. And so this works like that. So there's a 3-SNP window, there's a 3-SNP window, there's a 3-SNP window. And so we do this consecutively all the way through all 650,000 of those SNPs. And so this is what is generated after we do that. So this is a Manhattan plot. I'm sure all of you are aware of these sorts of plots. On the x-axis are the chromosomes. On the y-axis is the log p-value of the association between the haplotype and the phenotype. And all the points that you see are the 3-SNP windows that I just explained to you, and the association of the phenotype with that 3-SNP window. And as far as SNPster is concerned, if you have a p-value of a log p of greater than 5, this suggests that there's a gene in that region, or it's significantly associated with a gene in that region. And you can see on chromosome 1, we have a very high association for SNPster, and it's over 8. And if you focus in more carefully at that one region on chromosome 1, which is shown here, we identified in this peak a gene called MARCO. MARCO is a scavenger receptor. Its name is macrophage receptor with collagenous structure. It's a member of the innate immune scavenger receptor family, a very primitive molecule that is very important in a number of immune functions. We asked whether deletion of MARCO in mice would lead to an enhanced disease or increased susceptibility to RSV, and in fact it did. So on the left panel are the monocytes in the MARCO knockout and the MARCO plus mice. Knockout are in the blue and the wild type in the yellow, monocytes on the y-axis. And you can see that there are significantly greater numbers of monocytes at 5 days post-infection in the MARCO knockout mice compared to the wild type. On the right panel are the number of PMNs, and this observation was even more striking. So at 5 days after infection in the MARCO knockout mice, where we expect to see a complete resolution, we see a very significant enhancement of the inflammatory response. We don't know if these are new inflammatory cells, that is new PMNs, or whether it's old, the PMNs that have come into the lung and have not been cleared. Likely it's the latter. So then we asked in this translational study, what is the role of MARCO in human RSV disease? And for that project, we've been collaborating with Fernando and his group at Infant for a number of years. And Fernando showed a slide very similar to this yesterday. where this shows some of the hospitals, some of the regions in which hospitals have been working with infant, and you can see that some of the regions are middle to upper middle class, and others are of low socioeconomic status. This will come into focus or into importance a little bit later. So in these studies, we have used clinical outcomes. There's one primary, which is O2 saturations less than 93%, and secondary outcomes are RSV titer and Th2 polarization. This graph shows you, or this figure shows you, the homology between mouse Marco and human Marco. Mouse Marco over here on chromosome 1, analogous or homologous region of chromosome 2 matches very well with chromosome 1 on the mouse. This is the structure here of the human Marco, and we found potentially important promoter polymorphisms in Marco, in human Marco, which we then tested using luciferase activity assays. When you look at this mutation in this NRF2 ARE-like region of the promoter, the mutation leads to a significant suppression of luciferase activity, and the promoter polymorphism also enhanced the response to RSV in these cells. So we then asked whether that particular polymorphism that we showed to be functionally relevant in vitro was important in vivo. So we genotyped two populations, two independent populations for that for the polymorphism in mild RSV disease cases and in severe RSV disease cases. And in each population, if the individual had one or two copies of the mutated allele, you had greater chance of RSV severity than if you had the wild type combination. And when you combine the two populations as shown here, the odds ratio is 1.6 with a p-value of 0.003, suggesting there is in fact an important role of Marco in severe RSV disease in these populations. So that's our mouse study, and I've only shown you a very little bit of the work that's been done, and I'd be happy to discuss more if there's time. So the second project that we've worked on is an in vitro model of RSV susceptibility. And this is a project that was undertaken by a postdoc in my lab, John Sensowicki. And he developed an in vitro system using lymphoblastoid cell lines that were developed from the HapMap program, and I'll tell you about that in a second. But these cell lines can be used to identify genetic factors that control the response in these cell lines to any number of environmental pathogens. And so this is a description for those of you who don't remember what the HapMap program is like. So the HapMap program cataloged variants across the genome in 300 humans. These are usually trios, that is child-parent trios. And you can use these lines to assess the genetic diversity among the different ethnic populations. We used one, which is the CEU HapMap, which is derived from Caucasian families in Utah. And these cell lines are lymphoblast cell lines that have been immortalized, and you can purchase them and grow them up and use them for studies like I'm going to show you next. So in this study, John grew up the cells and suspended them in serum-free media, infected them with RSV, and then returned them to the media and at various time points after we harvested them and measured a number of outcomes or endpoints, including microarray analysis, viral protein expression, mRNA expression, and so on. So, does basal RSV gene expression vary between the infected haplotype cell lines? So this is the range of responses that we got across 47 different cell lines. The donors are on the x-axis. This is the fold change in RSVG mRNA expression after infection. Important points to take from this is, like we saw with the inbred strains of mice, there is a significant inter-individual variation between donors in terms of their viral load or viral titer, or in this case, viral expression. Upwards of nearly 100-fold variation from the high responders compared to the low responders. And that's shown here. So in these insets, these are low responders, which have about a 4-fold increase, a mean of about 1.5, and the high responders close to 200-fold. And so there's, again, quite a range in variation in response to RSV. So we can use a lot of current, that is available, publicly available information that is known about all of these lymphoblastoid cell lines. There are gene expression databases for each of the cell lines that you can access. There are 5 to 6 of these. And then you can ask whether differences in basal gene expression correlate with and or predict the response to RSV or whatever challenge that you're using in your cell lines. And so we've done that. We've correlated or looked at the correlation between viral load and the probe intensity for each of the data sets. And our significance was based on 3 different criteria. There had to be a p-value of less than 0.01 difference. The fold change had to be, of the high and the low responders had to be greater than 1.2 or less than 0.8 in genes that were downregulated. And it had to be observed in at least 3 different data sets. And this is a volcano plot showing one of these data sets. And so these genes in the sort of pink rectangle are genes that were downregulated, and in the green are genes that were upregulated and associated with differential responsivity to RSV in the lymphoblastoid cell lines. We then took these gene transcripts and modeled the response to RSV across multiple regression for each of these different transcripts and came up with a 27-gene transcript model, which is shown here, which is highly predictive of the response to RSV in the lymphoblastoid cell lines. One of the genes in this model is MX1. So MX1 codes for the MXA protein. This protein interferes with viral replication in certain viruses. We found that there is a SNP, a missense mutation at position 1135 in the MX1 gene, and it leads to a loss of function in the gene. And we asked again, this is a correlation using the expression array data that I mentioned to you that's publicly available of MX1 across all of the individuals that were phenotyped for their response to RSV. It's an inverse correlation. So those individuals that have low RSV expression had higher MX1 expression, and those that had high RSV expression had low MX1 expression. We then asked, in the same populations that we've been working with Fernando, whether this polymorphism associated with disease severity in RSV-infected infants. And in fact, it did, much like Marco, if individuals had one or two of the mutated alleles, you had an enhanced response or a significantly greater response, in this case severe disease, compared to those that had the wild type allele. And in this case, the odds ratios were between 1.7 and 1.9. So we believe this in vitro model can be used in a way to help us understand more directly inter-individual variation in human populations with respect to response to RSV. And the final study that I'm not going to have time to talk about is one that was published just recently, and it was an investigation of a candidate gene, toll-like receptor 4, which you all know has been studied very intensely over the last few years for its response to endotoxin, but also in response to RSV. And I will cut to the chase very quickly. We, in these populations that Fernando talked about yesterday, and I mentioned briefly before, measured LPS levels in the homes of individuals that were recruited for this study. Endotoxin levels ranged from very low to very high. They were very low in high socioeconomic status areas and very low in high socioeconomic status and high in low socioeconomic status areas. And then when we genotyped these kids from these different areas for the toll-like receptor mutation at TLR 299 or 399, individuals that had the mutation and were in low LPS areas had a much greater proportion of severe cases of RSV disease compared to those that had mild disease. And if you were in a high LPS environment, you had a greater proportion of individuals that had mild disease compared to severe disease. So this gene-environment interaction is summarized, and this is one population, this is another population, is summarized here. So if you have a combination of high socioeconomic status and the mutation, you are very much at risk, upwards of 8-fold to severe disease. And if you have a low socioeconomic status in the mutation, you're in fact protected against RSV disease. So what does all this mean? We think that understanding the genetic basis of susceptibility to RSV disease will enable us at some point soon to develop a diagnostic tool to help us genotype individuals for specific alleles that contribute to differential susceptibility, and also incorporate environmental status so we can evaluate gene by environment interactions and identify individuals that could be at risk to severe disease, and then provide a strategy to protect them from RSV infection or the progression of disease. And with that, I'll stop and take any questions.